specific patterns obtained on the electrophoresis are then utilized for interpreting the relatedness of diversity of the samples tested. Novel SSR marker sets have been developed in citrus, chilies, mango, brinjal, mung bean, banana, sesame, pigeon pea, watermelon and finger millet. More than 2,000 varieties belonging to 34 crops have been fingerprinted. Once the germplasm has been evaluated and characterized, seeds and propagules are multiplied for its conservation. The Indian National Gene Bank was established at nbpgr to conserve germplasm in form of seeds, vegetative propagules, in vitro cultures, embryos and pollen. The Gene Bank is supported by the active partnership of other institutions and centers designated as the National Active Germplasm Sites. The Seed Gene Bank consists of compartmentalized cold storage modules in which seeds are kept in controlled conditions of temperature and humidity. Seed materials on receipt are inspected and cleaned. They are then subjected to germination test following international standards. Seeds that show at least 85% germination and good vigor are the only ones that are accepted for conservation. The qualifying accessions are dried to low moisture contents under controlled environment. The dried seed samples are labeled, packed and vacuum sealed in tri-layered aluminium foil pouches. They are then placed at a defined location in the gene bank. There are 12 long-term storage modules maintaining a temperature from -18 degrees Celsius to -20 degrees Celsius at nbpgr headquarters and medium-term storage modules maintaining 0 to 10 degrees Celsius at headquarters and regional stations. Nearly 0.4 million orthodox seed samples, including wild relatives of important crop groups such as cereals, millets, legumes, vegetables, fibers, forages, medicinal and aromatic plants, are conserved in the Seed Gene Bank. A number of economically important plant species such as banana, sweet potato, taro, garlic do not form seeds or are propagated vegetatively. In addition, some species such as mango, jackfruit, lychee produce recalcitrant seeds, which are not amenable to seed conservation as their seeds cannot be stored at low temperature and low moisture. These plant species are either maintained traditionally in field gene banks or through use of in vitro and cryopreservation techniques. In vitro conservation, that is, growing the plants in glass under defined nutrient conditions in an artificial environment. Tissue culture medium is prepared as per the requirement of the crop and sterilized using autoclave. Suitable explants like shoot tips, nodal cuttings or shoot buds are used for culture initiation and multiplication in a sterile environment using a laminar airflow. Cultures are stored normally at 25 degrees Celsius under artificial light conditions. Presently, cultures of germplasm of more than 2,000 accessions of tropical and temperate fruits, tuberous and bulbous crops, spices, and some threatened medicinal and aromatic plants are conserved in the in vitro gene bank at nbpgr headquarters and Thrissur. In vitro conservation offers short to medium term conservation. For long-term conservation, cryopreservation is the only method available. Cryopreservation is the storage of viable biological propagules at ultra-low temperatures using liquid nitrogen that has a boiling temperature of -196 degrees Celsius. At nbpgr, plant germplasm in the form of seeds, embryos, embryonic axes, dormant buds, meristems and pollen are cryopreserved. The explants are suitably desiccated by air dehydration or by use of certain chemicals called as cryoprotectants before they are placed in polypropylene cryovials. The cryovials are plunged in liquid nitrogen where it remains frozen till its use. The germplasm is stored in the vapor or liquid phase of nitrogen. nbpgr has 6 state-of-the-art cryotanks of 900-liter capacity. The uninterrupted supply of liquid nitrogen is ensured by using a large liquid nitrogen station where filling and monitoring systems are automated. More than 9,000 accessions of important plant species have been cryo-stored in the nbpgr cryobank. All germplasm that is collected, evaluated and conserved is systematically documented. The most important step is assigning a unique identity number to each accession. IC number for indigenous collections and EC number for exotic collection. nbpgr has developed online databases for easy access and retrieval of passport data and information on germplasm exchange, quarantine and conservation of PGR. Detailed information on germplasm characterization and evaluation are available in the form of crop catalogs, and all plant germplasm, exotic and indigenous, are available in plant germplasm reporters and crop inventories. To promote utilization of germplasm, seeds are routinely multiplied and distributed to various users for research and crop improvement purposes. Field days, diversity fairs and farmers' fairs are conducted for live demonstration of material for their utilization. nbpgr is also facilitating global research programs by undertaking field trials of international nurseries of important crops. The Human Resource Development (HRD) has gained importance due to the increasing PGR-related activities in the changing global scenario. More than 500 scientists and students have been trained at national and international level in over 100 training programs. nbpgr is conducting postgraduate and doctoral degree program in PGR discipline in collaboration with the Indian Agricultural Research Institute, a deemed university. nbpgr has a specialized library on plant genetic resources with a collection of 12,000 books, 3,000 volumes of journals and online access to several journals. The headquarters and regional station at Hyderabad have museums depicting the plant genetic resources activities for visitors to get an overview of nbpgr. Recognizing the importance of plant genetic resources having novel, unique, distinct and stable traits of academic, scientific or applied value that could be used in crop improvement, a plant germplasm registration committee was set up by ICAR. The responsibility of germplasm registration has been entrusted to nbpgr and nearly 100 to 150 applications are received annually from researchers. A state-of-the-art national containment facility has been established to handle imported transgenic planting material meant for research purposes and to prevent their direct contact with the environment. This facility is used for quarantine processing and molecular testing of all imported transgenic samples for specific transgenes, promoters, markers and terminator technology. nbpgr is designated as referral center for molecular diagnosis of transgenic planting material. Rapid PCR-based detection kits have been developed for commercial use in 5 genetically modified crops to meet the regulatory obligations. The Bureau has distinction of receiving Best Institute of ICAR Award for the year 1997 in recognition of its dedicated and meritorious service to the nation. Over the years, it has the honor of being guided and visited by many important dignitaries. The genesis of nbpgr has almost coincided with the advent of Green Revolution in India during the mid-20th century. The 21st century is the era of Gene Revolution, and nbpgr strives to keep pace with the changing requirements. Newer programs on allele mining, gene prospecting, genomic resource conservation, phenomics and bioinformatics would help in better utilization of the vast plant genetic wealth in our custody. nbpgr strives to serve the nation by serving the cause of plant genetic resources.